Okay, let's get started. Thank you for joining us today. My name is Rhonda Henshaw-Powell. I'll be your moderator, and I'd like to welcome you to today's webinar, hosted by our West Coast team, Dijon Hill, Kristen Douglas, Abby Munoz. We would love to invite you to listen to the presentation today. This is a pre-recorded presentation that we held some time ago with Dr. Stephanie Shiers and Professor Theodore Price from the University of Texas, Dallas, in the Center for Advanced Pain Studies. They did a lot of work on understanding pain mechanisms and used RNA-scope, and so they looked at RNA expression analysis of the peripheral nervous system tissue. And so I'm going to go ahead and play the presentation for you. As you're listening, please do pop in all of your questions into the chat box. We have our support team standing by to address them for you. And we'll have a few moments from Kristen and Dijon or Abby at the end of the presentation, so please stay online. A couple of housekeeping requests. Please do stay on mute, and please turn off your video, as this directly affects our streaming quality. And with that, we will proceed to the presentation. Thank you very much. Apologies for that. I believe there's no audio. Let me go ahead and reshare the presentation. Let's see.
Hi, thank you for that introduction. I'm Stephanie.
Please confirm, can somebody confirm that you can hear the presentation? Okay, thank you.
Today, Ted and I have split this talk into two separate sections. The first section I'll be giving, which will be over our ongoing work using RNA-scope to characterize and start defining sensory neuron subpopulations in the mouse and human DRG, or dorsal root ganglion. And then the second half of the talk will be given by Ted, and he'll be talking about some of our recent work looking at viral-mediated pain and how this relates to the ongoing COVID-19 pandemic. So we started this project about 2 years ago in collaboration with Merck. They contracted with us to start trying to define sensory neuron subpopulations in human dorsoroganglion, or the DRG, using RNA-scope. Our project had 2 sub-aims. The first part was to identify sensory neuron subpopulations in human DRG based on known histological markers. So I'll be going over in a few slides what these histological markers are, but these are things that are essentially known to label specific subpopulations in the rodent field. And we just wanted to know if those markers could also be applied to the human DRG. And then the second part was then to analyze their co-localization with known peripheral neuron drug targets. So these were drug targets. We've done about 30 targets now that Ted and the people at Merck compiled a giant list of these. Most of these were of kind of historical classic interest to the pain community, so things like the voltage-gated sodium channels, NaV 1.7 and NaV 1.8, as well as the TRP channels, so TRPV1 as well as TRPA1, and then some more recently identified peripheral neuron drug targets that show promise in the preclinical field. We wanted to know what their expression pattern looked like in humans. So we've done a lot of these studies and this comparison in both mouse and human DRG now. So before I start getting into the nitty-gritty of our data and how we did RNA-scope, I want to talk briefly in just one slide about why a study like this is so important. Why is it important to look at sensory neuron subpopulations in human DRG? And I think this message is really portrayed nicely in 2 papers. The first one is a paper published by Jeff Mogul in Nature Reviews in 2009, where he speaks about translatability issues between mouse and human. So compounds that are being identified in the mouse that are working really well and show a lot of promise that are failing when they reach the clinic. And he speaks about how these may be due to issues with species differences or even sex differences and a variety of other reasons. And he has this figure in this paper that I think has a really nice message. So on the y-axis here, this is the number of papers published in the pain field. And then on the x-axis is publication year. So this is ranging from the mid-1960s all the way to the early mid-2000s. And what he's plotting is the species model of organism of choice that was used to study pain. And you can really tell up until the 90s, the majority of papers published were being conducted using the rat as the pain model organism. And in the 90s, in the red bar here, or the red line here, you can see that there's this large upshoot in the number of papers all of a sudden being published using mouse as the model organism. And this is really coming around the time in the 90s of the advent of the Crelox-P system and the availability of these transgenic mouse lines. And Dr. Mogul points out that this was really important in terms of understanding pain neurobiology and genetic function or gene function, because you can knock out a gene or manipulate it in certain ways and then look for phenotypic changes in behavior. However, this may not have been the best step forward in terms of species homology. Can we actually say that the mouse sensory system is homologous to the human sensory system? A lot of these characterization studies had previously been done on the rat, but are we actually, can we actually say that the mouse system is the same? And in that sense, are we actually just studying mouse pain and not human pain? And that really brings into this really nice paper published in 2018 by Rostock et al., where they did a really similar study to us, where they looked at human versus mouse nociceptor similarities and differences, looking at TRK receptors as well as neurofilament 200. And they have a really nice quote in this paper in their discussion that I think sums up this message perfectly, why these types of studies are important. So I'll read that. Rodents will continue to serve as important and necessary model organisms for pain research. Therefore, it is of critical importance to investigate similarities and differences between humans and rodents regarding cellular and molecular aspects of sensory neurobiology in order to gauge what aspects of this research have a translational potential, and additionally to indicate differences that may explain shortcomings concerning therapeutic expectations. So again, this is speaking to translatability issues that may be stemming from these differences in gene expression profiles between mouse and human. And in the end, are we actually just looking at mouse pain and not human pain? So how do we define classically in the rodent field sensory neuron subpopulations? Well, one way is by neuronal size. So in the sensory neuron, in the sensory dorsal root ganglion, there are 3 different sizes of neurons. There are these large myelinated A-beta mechanoreceptors that convey information about touch. There are these small myelinated nociceptive A-delta fibers that convey fast pricking pain. And then there's these small diameter unmyelinated nociceptive C fibers, which result in slow burning or aching pain. So we can classify neurons by size, but we can also classify them based on their protein or gene expression profiles. And classically, the markers that have been used to label specific neuronal populations are shown here. So for our A-beta mechanoreceptors, it's classically used to label them with neurofilament 200. We also have this population of peptidergic nociceptors, which are these mix of the A delta and C neurons C fiber neurons that release neuropeptides, so things like substance P as well as calcitonin gene-related peptide or CGRP. And then we have a population of non-peptidergic nociceptors, which can be labeled with P2X3 receptor or IB4. However, this system is a lot more complicated than just 3 different sizes of neurons and a handful of different protein or gene markers. There are labeled lines or specific subpopulations of neurons that convey information about different sensory modalities that can be labeled with a variety of different markers. So the system can get really complicated. So our goal of our work was to use these known histological markers in combination with a variety of these other markers, these other drug targets of interest, to look at expression profiles in both mouse and human. So we chose to use neurofilament 200 for our A-beta population, CGRP for our peptidergic nociceptor population, and then we chose P2X3 receptor for our non-peptidergic population. We chose P2X3 receptor over IB4. IB4 is actually more commonly used in the rodent field to label these non-peptidergic nociceptors, but IB4 is actually a plant lectin that binds to a carbohydrate residue on the surface of these non-peptidergic neurons. However, there's evidence to support that IB4 does not work in humans, so we chose to use something that we could actually detect at the mRNA level using RNA-scope, which was the P2X3 receptor. So we chose to use RNA-scope in situ hybridization, which is kind of a newer version of the in situ hybridization technology created by Advanced Cell Diagnostics. We chose this for a variety of reasons, all of them very good. The first one was that the probes are designed to be species and mRNA specific. This is unlike antibodies, which are kind of notorious for having a ton of problems. They can be fixation sensitive, they can be non-specific, they could cross-react with other protein families, they could give a lot of background. They may also not see both species, and we're looking at both mouse and human. So it's really nice that we didn't have to worry about that using these probes because they're already designed to be species and mRNA specific. We also get really nice, robust amplification of signal with this technology. So we use the version 1 kit, which is, I think, one of their original versions of this assay. It has a 4-step amplification process. So if you see over here on the right, step 3 is the amplification signal. It's a 4-step amplification process. So you have this giant kind of billboard attached to your mRNA probe, which is attached to your mRNA, so you get really nice signal to noise. You can multiplex for 3 target mRNAs, at least with the V1 kit. I know they have other kits available now where I believe you can do up to 12-plex, but we chose to just do their version 1 kit, so we can label 3 target mRNAs at one time. And then it also shows a potential application of working with immunohistochemistry. So we have 3 target mRNAs labeled in 3 colors, and so we have a 4th channel open, our blue channel, to be used with immunohistochemistry. And so this is unlike classic in situ hybridization, where there's a lot of harsh chemicals and harsh treatment of your tissue, and you usually destroy your antigen or your protein epitope. So this is really just dependent on one step in the process. It's a pre-treatment step in the RNA-scope assay, which is your protease digestion. So if you just optimize this protease digestion step, and kind of find the sweet spot where you get really nice RNA scope signals and still maintain your protein integrity and still can get nice IHC label. It's just a really simple part in the process. So on the right here, we have a 4-channel colored image. There is RNA labeled in green, red, and white. And then we have blue staining, which is our Neurofilament 200 IHC. And then I think the most important thing about the RNAscope technology, which really sold us on using this for our human studies, was this internal mRNA quality check that comes with the kit. This is really especially important for working with human tissue, which you're not always sure about the conditions of the tissue you're going to receive. How long was the body deceased for before the tissue was collected? Was it frozen in media? Was it frozen on dry ice? Was it frozen in liquid nitrogen? Was it fixed for a week? You know, all of these conditions can definitely affect the quality of your mRNA. So gauging that the quality of your mRNA before you even start these kind of time-consuming and expensive studies is really important. So we did this mRNA quality check for every sample we worked with. This is how it works. So it's a quality control, positive control cocktail. So the cocktail contains 3 housekeeping mRNA, or probes against 3 housekeeping mRNAs that vary in their degree of expression. So on the right here, you can see a low expressing housekeeping mRNA, a medium, and then a high. And then you can also, using this, optimize that protease digestion to try to get that nice IHC label, which we're showing here with Neurofilament 200. This is a mouse dorsal ganglion. And so the idea here is that you really want to see signal in all 3 of your mRNA target probes. And you want to see this kind of varying degree of expression, because the idea is if you only see the medium expressing transcripts as well as the high expressing transcripts, you may be missing some low expressing or detection of your low expressing genes when you do the RNAscope assay. And this is what this looks like in humans. So we have our high expressing transcript. I also do want to point out, so this is a 20x image of a human DRG. Oops. And then we also have a 20x image of our mouse DRG. So in the mouse DRG is so much smaller, we can capture the entire thing in one 20x view, but we can only capture a very small portion in the human DRG. So I just wanted to point that out. So this is a high expressing transcript, our medium to high, and then our low expressing transcript. So we only worked with human or mouse tissue that we saw expression for all 3 of our housekeeping mRNAs because we were doing kind of an exploratory study, which we didn't know what these gene expression profiles would look like in these tissues. Now, I do want to point out, since you'll be seeing some background label in our background in our human samples, what background looks like in human nervous tissue. So human tissue is fraught with issues, mostly background issues, and that's why people are hesitant to work with them. So a lot of the green signal you're seeing here is autofluorescence from fat. So this is myelinated fibers with high water content. So they autofluoresce a lot in green. And then you can see in white, this is these large globular structures. These are actually lipofuscin. This is known as the wear or tear pigment or the age pigment. These accumulate in nervous tissue and in neurons as we age. And they can be problematic, especially if you're doing protein staining and looking at lysosomes or some other type of protein that might give a similar signal. However, for us, it wasn't a problem. For one thing, the lipofuscin autofluoresces in both the green, red, and far red channels. So in the overlay image, you can see that these appear white. So it's really easy for us to rule out that this is not a real RNAscope mRNA signal. The size difference is also a good indicator because the mRNA puncta is much smaller than these, even the smaller lipofuscin puncta. Okay, so to get into the data, so of course we first started with these known histological markers. We use CGRP and P2X3, RNAscope, and C2 hybridization. So these are mRNAs, so I am calling them by the protein name here just for simplicity. And then we have immunohistochemistry for Neurofilament 200. And I think you can appreciate in the overlay image, that there are green cells that look entirely green, there are blue cells that look entirely blue, and then there's cyan cells that look very cyan. So these look like very distinct populations in the mouse DRG, which is similar to what's been reported before. And when we quantify this, and we just look first at the CGRP and P2X3 expressing neurons in this pie chart, you can see that a huge piece of this pie is P2X3 positive only, or CGRP positive only. And then there's a smaller piece, around 15% of the neurons, that co-express both CGRP and P2X3. And so on to the left, you can see that these are mostly smaller diameter neurons, around 20 to 30 micrometers in size, which is really similar to what's been reported in the rodent literature previously. Of course, when we bring neurofilament 200 in this picture, you pretty much get every combination you can have of these different subdivisions, but I think you can appreciate that there's a large piece of this pie, around 30.4%, that only expresses neurofilament, around 32.6% that only expresses P2X3, and then around 11% that expresses CGRP only. So these are still pretty distinct populations. And then on the left here, we're now including the neurofilament population into this histogram, which are all skewed to the right. So you can see that these neurofilament positive neurons, which remember are these myelinated large diameter A-beta mechanoreceptors, they're very large in size, which is definitely matching with our data. So what about humans? That's the question. So we knew when we started the study, literally this paper came out the same month we were starting these experiments, that neurofilament 200 wasn't going to work for us in human, because this paper by Rosak et al., which I mentioned earlier, published data where they stained for neurofilament 200. This is IHC staining in human and mouse DRG using two different neurofilament 200 antibodies. And on the right panel is actually the same antibody that we're using in our mouse studies. And what they found was that in mouse, neurofilament 200 labels these large diameter neurons very exclusively, similar to what we found. But in human, it actually labels all of the sensory neurons. And me starting these experiments and not having read this paper yet, I also did the same type of experiment where I stained human DRG with neurofilament, this panel on the right in cyan, and I saw neurofilament 200 staining in all sensory neurons. So right off the bat, we have a specific histological marker in mouse that's no longer working in human. So in lieu of using neurofilament 200, we just used DAPI. This allowed us to use the longer protease digestion on our in situ hybridization assay for RNA-scope. And so you can see CGRP signal and P2X3 receptor signal here. And I think in the 100x image, you can see this even more, is that there's a large percentage of neurons that are now co-expressing CGRP and P2X3. These aren't as exclusive of populations as they are in mouse. In human, they look to be very much co-localized. And so you can see now in this pie chart, this purple chunk here, which used to be in the mouse exclusively P2X3 positive, now looks to be CGRP and P2X3 positive. And these neurons are small in diameter, so these are around 50 micrometers in human. So when we put these pie charts side by side, I think you can see now that this CGRP and P2X3 population is much larger, co-expressing population is much larger in human than it is in mouse. And we wanted to figure out, like, what's the main cause of this? So what we did was we graphed the percentage of neurons expressing CGRP in human and mouse, and you can see that there's a lot more neurons in human expressing CGRP than in mouse. So around 30% in mouse, and it's now up to around 60% in human. The difference between P2X3 expressing neurons is there is no difference between mouse and human. So really the culprit for this larger CGRP P2X3 expressing population is due to more neurons expressing CGRP in human than mouse. So all of these things are bringing into question, not only do we have neurofilament 200 being expressed in all sensory neurons, we have differences in CGRP and P2X3. So this is really blurring these lines of what are these labeled lines. So I do want to talk briefly about TRPV1. This is a kind of classic pain marker that the pain community has been interested in quite some time. This is the capsaicin receptor, responsible for heat-mediated pain. And I do have some data on mouse. So this is CGRP, P2X3 receptor, and TRPV1 labeling in mouse DRG. And then Neurofilament 200 is added in the far right panel. Let me move on so you can see this zoomed in a little bit more. So you can see that there are a lot of yellow cells in the overlay image. So this is CGRP and TRPV1 co-expressing neurons, but there's also a small population of neurons that co-express P2X3 and TRPV1, and this is considered to be probably the itch population. And if you look at the pie chart, TRPV1 is expressed in around 30% of the cells. So these are a really small population of neurons, and they are also small diameter, as shown on the left histogram. So this has also been shown at the protein level. So if we do protein staining for TRPV1, it's in around 30% of the cells as well. So the mRNA and the protein data appear to be matching in mouse. So what about humans? So I have a mosaic image that has a zoom in. So this is CGRP signal. Then we have P2X3 signal. And then we have TRPV1. And I know that was kind of fast to click through there, but I think you can notice that virtually all of the neurons that express CGRP or P2X3 are also now expressing TRPV1 in human. And if we zoom out, you can see all of these neurons in the human DRG that are expressing TRPV1. So there's a lot more neurons that are expressing TRPV1 in human. When we quantify this, we see it in around 75 to 80% of the neurons are expressing TRPV1. It's in virtually all of the CGRP and P2X3 expressing neurons, and these are also small diameter in size. So if we graph this and quantify this, so if you look at this as the percentage of neurons that express CGRP and P2X3 and what percentage of those neurons co-express TRPV1, it's in around 90% of those nociceptor CGRP P2X3 positive neurons. So this is in virtually all of these nociceptive neurons. And when we do the statistics and compare mouse versus human, we're seeing much more TRPV1 in human than in mouse. So in mouse, it's around 30% of the neurons, and in human, it's around 75 to 80% of the neurons. So this is very much different than what's been in mouse. So I'm going to change pace a little bit and talk about some other interesting things that came out of us using RNA-Scope, and that is looking at different subcellular localizations of the mRNA signal. So I'm not showing it here, but we also published in our recent paper, actually just came out in Pain last week, that accepted. So if you want more details on the RNA-Scope assay and our methods, we have very detailed methods section of how we did these experiments. But we also have a lot of data. So there's 17 different targets we looked at in that paper. One of them was HDAC6, which actually is a histone deacetylase 6, and it showed at the mRNA level a very interesting subcellular localization where it was only found in the nucleus, or the majority of the signal was only found in the nucleus. And similarly, we also could see really unique subcellular localization patterns with the mRNA for a variety of targets. So this is HCN1 and SCN9A, or the NAV1.7 gene. And we could see mRNA signals showing up in axonal fibers. These, interestingly, both of these targets are actually expressed in all sensory neurons at the gene or at the mRNA level in human DRG. And if we look at just confirmation of this work at the GTEx consortium, where they have sequenced a variety of different human tissues, we see HCN1 signal and SCN9A signal in the tibial nerves of human patients. So this suggests that this is a real signal and we're actually detecting mRNA in these fibers. We were also able to look at protein as well. So we had a really nice NAV1.7 knockout validated antibody that worked on human DRG. So this is NAV1.7. So the previous slide showed SCN9A mRNA signal, which is the gene for this protein. And you can see that there's a lot of Nav 1.7 IHC label in the DRG on the left-hand side in this bulb-like structure. It's in around 90% of the neurons. I think you can appreciate there's these really large cells that aren't expressing Nav 1.7. Those are probably the A beta population. But we also see a lot of Nav 1.7 protein in the fibers as well. So this is matching our mRNA data pretty nicely. And we also have some preliminary work that we've been doing where we have tibial and sural nerves collected from patients with diabetic neuropathy who are undergoing below the knee amputations. And so we've been doing some molecular studies, some sequencing, and some RNA-scope to see if we can detect mRNAs that we predict would be expressed in these nerves. So I've done some preliminary studies looking at TRPV1 as well as that Nav 1.7 mRNA using RNA-scope, and we can detect these mRNAs in the nerve. And we've been looking at the protein as well, and we're getting similar results. Also, in our recent paper, we did look at some non-neuronal mRNAs. So we looked at CD68. So this is kind of a classic marker used to label macrophages, but it also labels a variety of other cell types, including fibroblasts. And I think you can appreciate just from this left-hand picture that the CD68 signal is making this ring-like or halo-like shape around the sensory neuron. And based on this localization around the sensory neuron, we predict that these are satellite cells, which are labeled on the right. So this is GFAP labeling of satellite glial cells in the human DRG. And these are making these ring-like shapes around the neurons. And these satellite glial cells, I was very surprised, are very big in human DRG. These are around 10 to 20 microns in size. So very similar in size to even the small mouse neurons. And recently we started looking at human spinal cord. So we actually collected tissues from our first human organ donor that we were actually able to collect tissues ourselves from. So right before we started quarantine, we were able to get some human spinal cord, and I ran some positive control quality check, which gave us really nice RNA-scope label with the positive control. And I did some protein staining to look at Nav 1.7 because we knew that antibody gave really robust staining in the fibers. And the mRNA was found in the fibers as well. So when I did human spinal cord Nav 1.7 staining, if you see in the top left here, you can see part of the dorsal rootlet coming from the DRG and all the Nav 1.7 labeled fibers coming into the spinal dorsal horn and giving this neuropill-like pattern in lamina 1 and lamina 2. So this was really exciting for us to start trying to tease out where these fibers might be synapsing into the dorsal horn of the spinal cord and start trying to do these RNA-scope and immunohistochemistry studies. So I'm going to let Ted take over now and talk about our work with viruses and pain.
Thanks very much, Stephanie, for that fantastic talk. And I will take over and actually going to talk a little bit about some of Stephanie's additional work that she didn't talk about in her section. And I'm going to focus mainly on work we've been doing to try to understand how viruses might be involved in causing pain. So I'm going to talk about this paper that was published recently in Journal of Neuroscience. And then if you're talking about viruses nowadays, you have to talk about COVID-19. And we've been doing a little bit of work on that. So I'll also wrap up by talking about some of our recent work on potential neuroimmune interactions and virus and sensory neuron interactions with the SARS-CoV-2 virus. So I'll talk first about this paper. So we are interested in lots of different mechanisms involved in pain generation. And I think most people are aware that one of the first symptoms that most people get when they have a viral infection is body-wide aches and pains. And those body-wide aches and pains often come about before people get a fever. So it's unlikely that they're driven by the fever necessarily. And we know from lots of work done by many other laboratories over decades now that one of the first responses that your body has to a viral invader is to increase production of type 1 interferons. So the question that we asked in this paper, which I'll take you through the data, was whether or not type 1 interferons might act directly on sensory neurons. So we used mice for these studies. And what we did at first was just simply to use naive mice, male and female mice. You can see male mice on the top, female mice on the bottom. And we gave them injections of either interferon alpha or interferon beta, which are the two major type 1 interferons. And you can see that injecting these into mice causes a fairly long-lasting mechanical hypersensitivity. Interestingly, they don't seem to cause thermal hypersensitivity, so that they're relatively selective, at least in mice, for the mechanical pain pathway. So that was the first indication that interferons are potentially part of the initial pain response to viral infection. So we then wanted to understand, this is work that Stephanie did, whether or not these receptors were actually expressed by mouse DRG neurons. There's two primary receptors for type 1 interferons. The interferon alpha or type 1 interferon R1 receptor and type 1 interferon type 2 receptor. And these act as a heteromer to induce signaling. So you need to have both of them. And you can see here that most mouse sensory neurons do express both of these. So it's plausible that type 1 interferons could act directly on mouse sensory neurons. So then there are a number of different signaling pathways that have been identified for type 1 interferons. These include ERK signaling to EIF4E, which is a RNA binding protein that regulates translation. Another one is the AIF2 alpha signaling cascade, which suppresses cap-dependent translation, and then the STAT signaling pathway. We observed that in sensory neurons, at least, we didn't see any activation of the AIF2 alpha pathway, but we did see transient activation of ERK signaling and EIF4E phosphorylation with both interferon alpha and interferon beta. So they seem to be able to act on sensory neurons. These are cultures of sensory neurons from mice, and this actually has a mixed cell population. So we wanted to look at single cells to see if their excitability was changed by in type 1 interferons. So here what I'm showing you at the bottom is a patch clamp electrophysiology experiment where we exposed dorsal root ganglion neurons to either a control solution and then hit them with a depolarizing ramp current, which makes them fire action potentials, or interferon alpha and then hit them with the same ramp current. And I hope you can easily appreciate here that interferon alpha makes these neurons quite hyperexcitable. So that's a really good indication, again, that type 1 interferons can act directly on sensory neurons, and the consequence of that is activation of ERK, EIF4E signaling, and an enhancement of excitability. So we wanted to look a little bit more precisely at the signaling. So we used cultured neurons again and did immunofluorescence. In this case, we used two different markers, peripherin and NUN-N. So NUN-N is a general neuronal marker. Peripherin is a marker of mostly small diameter sensory neurons. And you can see from the vehicle to the interferon alpha and interferon beta exposure that EIF4E phosphorylation clearly goes up, and it goes up in the peripherin positive neurons. So this, again, is consistent with the idea that these type 1 interferons can act directly on sensory neurons to induce signaling and increase excitability. So we have worked actually for many years on ERK, MINC, EIF4E signaling in the context of pain, and we have knockouts for MINC, which is a kinase that's in between ERK and EIF4E phosphorylation, and then we have these mutant mice that have a point mutation that renders them unphosphorylatable by MINC in EIF4E. So basically, they're phosphorylation null animals. So we wanted to then test whether or not type 1 interferon-induced mechanical hypersensitivity would be abrogated in these mice. So what you can see on the left is that when we expose MINC1 knockout mice to interferon alpha, that they basically don't show any mechanical hypersensitivity. Same story in the middle with interferon beta, doesn't induce any mechanical hypersensitivity. And then finally in the the EIF4E phosphorylation null mice, again, type 1 interferons fail to induce mechanical hypersensitivity. So we concluded from this paper that indeed type 1 interferons can act directly on sensory neurons. We think that this is a really good explanation for why viruses cause body-wide aches and pains, and that's one of the first symptoms that you get. The virus induces type 1 interferon. Sensory neurons throughout your entire body are tuned to respond to the type 1 interferon. It induces signaling through MINC EIF4E phosphorylation, and that then drives hyperexcitability in nociceptors to cause viral-induced pain. Now, there may well be other mechanisms that are involved, but we think that this is a good explanation for at least some of the sensory effects that are caused by viral infection. So now I'm just going to quickly talk a little bit about work we've been doing on COVID-19 and the sensory system. So I think most people recognize that your skin and your muscles and your bones are innervated by sensory neurons, but in fact, almost every organ in your entire body is innervated by sensory neurons. And these sensory neurons can do some pretty surprising things. You know, we know that they can be involved in our detection of light touch, or not that they're involved in it, they mediate our detection of light touch. They play a critical role in pain and detecting things that can hurt us. They play an absolutely crucial role in the development of chronic pain. But they also do things like regulate the immune response within our organs, such as our lung. So in the context of COVID-19, we wanted to try to understand whether or not sensory neurons were playing a potential role in the response to the disease. So we were in a good position to do this because we've been working with human dorsal root ganglion neurons for a long time. And we actually have been profiling thoracic human dorsal root ganglion neurons for quite some time through a collaboration with MD Anderson, where we're getting thoracic dorsal root ganglion from patients that are having a surgery called a vertebrectomy. So it turns out that the lungs are innervated by two primary sensory sources. One source comes from the vagus nerve. We don't have any access to vagal afferents from humans. The other is from the thoracic DRG. So we have good data sets, RNA sequencing data sets for these neurons. And we also were lucky to spot some papers that were published pretty early on in the pandemic, where some investigators in China were taking patients that either had minor or severe COVID-19 and extracting cells using bronchoalveolar lavage, or BALF, and then doing RNA sequencing. And these investigators were kind enough to share their sequencing data sets with us really remarkably openly after they had either published them on medRxiv or bioRxiv. And basically what we did is that we just took the sequencing information that we have on human DRG and looked at the receptors that are there, and then assessed the ligands that are produced by the immune cells in people that had COVID-19, and looked for potential ways that the immune system could be interacting with the sensory innervation of the lung in the disease. So this paper has now been published in Brain Behavior and Immunity, so you can go find it if you're interested in reading all the details. But essentially, what we found was that these immune cells and they're mostly macrophages, but these immune cells that are in the lungs of the people with severe disease are increasing a set of chemokines and other ligands that have receptors that we think are fairly robustly expressed by dorsal root ganglion neurons that innervate the lung. And three of the most highly increased inflammatory factors coming from these immune cells are CCL2 and epiregulin. And CCL2 has a number of different receptors that are expressed by human dorsal root ganglion neurons. And then the epidermal growth factor is expressed by sensory neurons. So we think we've pinpointed a number of different interaction points here that we could potentially interfere with. And one thing that's kind of interesting about this is that a number of the drugs that can interfere with these pathways are used for rheumatoid arthritis. So we kind of concluded in this paper that there are a set of drugs that are currently used to treat other diseases that could potentially be used to treat severe COVID-19. So of course, we don't know if that can actually be the case until the clinical trials are actually done. But I've been impressed to see that a number of clinical trials are now happening around these kind of drugs. So I guess we're going to be learning more in the months to come. Sadly, the pandemic doesn't seem to be slowing down, at least in the United States. So there will be plenty of opportunity to do these clinical trials, unfortunately. So one other question that we kind of we had based on these initial studies, which was shown in a speculative fashion on the summary figure from that previous paper, was whether or not the virus might actually infect sensory neurons themselves. And I think most people now know that the virus gets into cells by acting on this receptor ACE2, the angiotensin converting enzyme type 2. And so we simply wanted to ask the question, do sensory neurons express ACE2 RNA? So Stephanie and I set out to look at this using RNAscope, again, as Stephanie talked about earlier. We used thoracic DRGs to do this because we wanted to look at DRGs that are potentially innervating the lung. And what we found was that indeed a subset of cells in the human dorsal root ganglion do express ACE2 mRNA. All of them are smaller diameter neurons. Most of them express CGRP and P2X3, as Stephanie mentioned. In humans, these overlap really strongly, whereas in mice, they don't. And interestingly, these neurons also expressed two other markers, MRG-PRD, which is a mass-related G protein coupled receptor, and beta natriuretic peptide, or NPPB, which is a neuropeptide. These are important because these markers are expressed by neurons that innervate the outermost layer of your skin and also innervate your hollow organs. And that they tend to form free nerve endings. So this could suggest that the free nerve endings that are found in the lung are likely to be expressing ACE2. So right now what we're trying to do is trying to better understand whether or not the protein is expressed. The mRNA expression for ACE2 in human DRG neurons is admittedly not very high, but it's certainly detectable, and we don't really know much about how mRNA expression equates to protein expression for this particular gene. So I think we have a lot to learn there, and Stephanie's actively working on those experiments. So I'm going to wrap up and we'll take questions, but I just want to point out all the really great people that work in my lab that have made this work possible. Of course, Stephanie, who gave a really fantastic presentation in the first half today and contributed to most of the work that I talked about in my talk. Paulino Barragan Iglesias is not on the slide because he's now left my lab, but he did the interferon work, and he's now started his own lab in Aguascalientes in Mexico, and I look for really great work from him, as he's just had a fantastic level of productivity and innovativeness when he was working in my lab. And also want to point out Perdita Ray, who does the sequencing work that I talked about. He's the first author on the BBI paper. And Andy Wang Zhu and Cam Page, who have put together this interactome framework that allowed us to do the work that I described in the BBI paper. Finally, we had really great support from Merck on the human DRG project. And also, we just simply wouldn't be able to do anything were it not for the really great support that we have from NIH and the National Institute of Neurological Disorders and Stroke. So I will stop there, and we're happy to answer any questions.
Great, thank you very much, Ted and Stephanie, for a very interesting presentation. It's now time for our question and answer session. So you can submit your questions, just click on the tab to the left of your screen and you'll see the part to submit your question. So the first question we have here: how do you deal with lipofuscin in human samples when quantifying signal?
So I touched on this briefly about how the lipofuscin signal autofluoresces in the green, red, and far red channels, so it's really easy to rule out because it looks white in an overlay image. Or you can just toggle between channels and tell that it's not real signal. However, I didn't mention that we actually did try to block the lipofuscin with some known treatments that you can use post-treatments. So I have a lot of experience using Sudan black, which is used to turn essentially the lipofuscin black. The lipofuscin is still there, but it's colored black, so you can't detect it in your fluorescence imaging. However, the problem with using that type of treatment is that it also turns kind of your entire sample dark, which can reduce the fluorescence intensity of your real signal. So when we're looking at really tiny puncta or even puncta for mRNAs that are very lowly expressed, it can be problematic because we may just be getting a bunch of false negatives because they might actually be there but just be reduced in intensity due to the Sudan black or the True Black post-treatment. I actually have not tried True Black. I've heard good things about it, so that is something we could look into. But other than that, we actually used it to our advantage, lipofuscin, because it is in the neurons, so it's easy to see the neurons that way, and it's easy to rule out because it does autofluoresce in all three colors.
Great, thank you, Stephanie. The next question: you show species differences between mice and humans. Are rats more similar to humans in the molecular features of the nociceptor subtypes?
Stephanie, you want me to take that one?
I mean, it's your paper, so yes.
All right. So I think so. So we actually published a paper when I was a graduate student on differences in CGRP and IV4 positive populations between mice and rats in the dorsal ganglion and in the trigeminal ganglion. And the short summary of that paper is that in mice, as Stephanie described, these two populations are pretty segregated. In rats, they're not, but they're not still as overlapping as they are in humans. So I think the rat is somewhere in between. I think that doing probably a single cell sequencing experiment would be the right thing to do on rats. Single cell sequencing has been challenging in humans, and I'd be happy to talk about that, why that is, if somebody has a question about that. But I think, DRG single cell sequencing experiment on rats would go a long way to help us understand at a global level how the populations of sensory neurons differ between mice and rats. And eventually, when we get to the point where we have that kind of high throughput data for human as well, then I think we'll be able to do quite a bit. Okay,
thank you, Ted. And to our audience, if you do want to submit a question, you can do that using the tab to the left of your screen. So the next question here, do you think that prolonged exposure to type 1 interferons may be a cause of neuropathy in development after viral infections?
Yeah, that's a great question. Paulino is going to be working on that in his laboratory. He got a grant from the International Association for the Study of Pain to do exactly that, and I'm completely confident that Paulino is going to have some really interesting results from what I think will be a really cool study. So, terrific question, and look for Paulino to be publishing some great papers on that to come.
Thank you, Ted. The next question, when you talk about mouse expression, what mouse strain are you talking about?
So we use the C57 Black 6 strain. We used males and we used young mice. So like I pointed out in that Jeff Mogul paper with that figure, a lot of these mouse papers that are being published nowadays are using transgenic mice, which have a C57 Black 6 background. So we wanted to be consistent with that. We are also using younger animals. So these may not be the best representations of human DRG or the most homologous of tissue types or species types to choose. This is an inbred strain of mouse. We are looking at males only, and we are looking at younger mice, which is similar to what the field has used. So this may not be the best reflection of the human DRG. However, this is what is, I think, more robustly used right now in the field to study pain.
And I'll just add one thing to that. We have done, so in this particular study, Stephanie didn't do any direct comparisons with male and female, but we have done many studies using female mice and looking at CGRP and IB4 co-localization and different neuronal populations, and we haven't seen any sex difference in terms of the neuronal populations, at least in the Black 6 strain. I'll also just say that we have done similar studies in the past, again, not part of Stephanie's study here, but where we've done CGRP and IB4 and outbred strains, et cetera, of mice. And we've never seen considerable overlap between the two populations in mice. So I think the segregation of the IB4, the non-peptidergic and peptidergic in mice is likely to be true in males and females and in different strains. Now, the age question is a great one. I haven't seen anybody address that systematically, and we certainly have not.
Great, thank you both. The next question we have here, how do you quantify high and low levels of mRNA expression, which can differentiate between different types of neurons?
So in the first paper, we did not do fluorescence intensity signal analysis. I just analyzed it. If it had a single puncta for a specific mRNA target, it was labeled as a positive neuron. So that is true. It could be, there could be high expressors or low expressors or medium expression for the same RNA target, but we didn't do that type of analysis. We are working out a way to kind of try to measure abundance using area of the neuron that is covered with RNA or with punctate signal to see if we can see any differences that way. However, that type of analysis is really time consuming and a lot more difficult to do than just labeling it if it's positive or negative. But that's still ongoing right now, something I'm actually working on.
Great, thank you, Stephanie. The next question, are there differences between human subjects, age, sex, medical history?
We are trying to address that now, mostly through this MD Anderson study. So we had a paper that we published in Brain last year that had a mix of electrophysiology and RNA sequencing on pain versus non-pain samples from people having these vertebrectomy surgeries. At the time, we didn't have enough samples to be able to say something about age or medical history other than whether or not they had pain. but I would say that we're rapidly getting to that point. So I think I mean I can't answer the question right now, but we're approaching it, and we have close to like 60 human DRG samples that we sequenced, where we have pretty detailed medical histories and even some electrophysiological characteristics. So I'm pretty excited about what we'll be able to do in the relatively near future in that regard. And the other thing that's important is we have another banked sample for each one of those individuals, so we can go back and do this kind of detailed cellular analysis on individual patients using the approaches that Stephanie talked about today.
Thank you. The next question, do you know if a NAV 1.9 channel participates to pain and inflammation pathways in SARS-CoV-2?
I don't know. I think we don't even yet really know whether or not SARS-CoV-2 is directly contributing to any kind of pain phenotype. I mean, there are some really interesting things out there. So one is that one of the early symptoms that people get is loss of taste and smell. But there was a really nice paper that was published in medRxiv fairly recently that will probably be published in a nice journal here relatively soon, showing that people also lose chemical sensation in their mouth when they, or many people do when they have been infected with the virus. And that is at least partially mediated by nociceptors from the trigeminal ganglion, which might also express ACE2. So that's one important thing, I think. And then I've heard from and also read in some kind of review papers, but nothing at, I would say, like the full-blown epidemiological study type level, that people that have pre-existing neuropathies get worse if they get COVID-19, and I guess we don't know at this point how long that will last. A lot of patients complain of pretty bad pain, but we don't have any idea what the mechanisms are and if they involve NAV 1.9 or not. We could look, I think, to see whether ACE2 is expressed in the population of neurons that also express NAV 1.9, but we haven't done that. I can tell you that they definitely express NAV 1.8 and NAV 1.7 because it's a nociceptor population, and in humans, all the nociceptors or putative nociceptors express NAV 1.8 and NAV 1.7.
Thank you, Ted. We've got time for one final question. Is N equals 3 used in human expression studies enough to address variability between samples and patients?
I didn't hear the question.
So
the first part. Could you repeat the question?
Is N equals 3 used in human expression studies enough to address the variability between samples and patients?
No, but it's hard to get enough of the tissue. So I think eventually we'll be able to do this at a much larger scale. But I can say, and Stephanie can comment too, I mean, one thing that has impressed me in the RNA scope work that we've done, there's remarkably little variation from organ donor to organ donor. So most of the work that Stephanie talked about is DRGs that we've collected from organ donors, not from these patients from the MD Anderson study. So I have been impressed that the variability in the neuronal populations is relatively small. So I think we can get a good idea about that. Now, if you're talking about genes contributing to neuropathic pain phenotypes in the patients, I think that's going to be a lot harder to do with a relatively small N, which is why we've been building these data sets up as much as we can. Our goal in our grant that just got funded by NIH is to have 160 patients enrolled over 5 years. Given that we've already enrolled about 70, I'm pretty confident that we'll be able to hit that that number, and I think we'll be able to learn a tremendous amount from what we do, but we're really just scratching the surface on that project right now. So we have a lot of work to do.
I think too, the low variability is just really mostly dependent on the RNA-scope assay and the quality of the mRNA in the samples. If you look at some of the classic in situ papers with N sample sizes of like 10 to 12 human DRG samples in it, there's a lot of variability. And I'm guessing they had to use a large N because they were getting some bad quality tissue. With the RNA-scope technology, as I went over with the positive control, we can gauge the quality of the mRNA. And I think that's really helped us with getting these really nice low variability data sets. We've also, so I only showed an N of 3 for a lot of these different targets, but we now have probably 6 or 7 human DRGs. And for all of these, the histological markers we've been using, we're not seeing any differences. And we have a wide range of ages, race, sex, from patient to patient from these donors. So we're not seeing a ton of variability, but like Ted said, it could be gene specific, and then haven't looked at them yet.
Yeah, so just one more quick comment on that. I think it's also important to note that a lot of the papers that were published previously were using post-mortem tissue, which you would expect there would be a lot of variability in the RNA quality. And Rob Giroux's lab and the folks at ACD-Bio, I think, completely changed the way that we do this by opening up this idea of doing it from organ donors. So basically, right after the organ donation surgery is over, you go in and take the DRGs and spinal cord, and that allows you to have really pristine mRNA quality in most of the samples that you obtain. So I think that's a pretty much a game changer, and the credit there totally goes to those two groups, or Rob Giroux's group at WashU and ACD-Bio.
Thank you both. That's all we have time for today. So thank you very much to Ted and Stephanie for today's informative discussion and presentation. And thank you to everyone joining us online and for your interesting questions.
Thank you very much. That was an excellent presentation. Like you say, we have been able to address most of your questions as they come through, but if you have specific questions for the speakers, we will reach out to them and have them come back to you. I hope you enjoyed it today. I'd like to just hand over briefly to Abby. She'd like to say a few words on behalf of the West Coast team. Abby.
Yes, so hi everyone. Hope everyone's doing well. Thanks for joining the Pain Mechanisms webinar. I hope you enjoyed it and got some information, some informative data presented by Stephanie Shiers and Dr. Price. I'm Abby, the RNA-scope experiment consultant. I cover SoCal, Arizona, Utah, Nevada, and New Mexico. My colleagues Kristen and Dijon are also on the call, and Kristen covers Oregon and Northern California, and Dijon covers Washington and Colorado. I can tell from the call I've been really lucky enough to have met and spoken with a few of you, and we all would be happy to chat with you all to learn more about your research and possible experiments where you see RNA-scope as a good fit for answering your research questions. And I've learned also from the pain researchers that I've spoken to, and I'm sure Kristen and Dijon have spoken to, that the IHC application is very important when understanding diseased versus non-diseased models. So we're happy to announce that we have a new code detection kit available for enabling a robust IHC data. And it has very minimal background to no background. So we're very excited about this new launch. So if you guys are interested in that IHC dual application, please reach out to us. We'll put our information in the chat box and just let us know if how we can help create a good environment for you guys to learn more about your research, and we're happy to help you guys out.
Great, thank you so much, Abby. And again, thank you, Kristen, Dijon, and Abby for hosting such a wonderful presentation. We are definitely very excited to share this work with us, and we're definitely excited for the new webinars and the new products coming forward. So please do check out the events page at acd-bio.com. Register for some more upcoming webinars, specifically for the co-detection webinar launch is actually September 29th. And so if you can go ahead and register for that, I'm sure you'll learn a lot. I know that Dr. Shiers and Professor Price mentioned that workflow indirectly in their research today. So if you want to learn more, please check it all out on our website. This webinar has been recorded, so we will make the recording available online on our recorded webinars page in the next 24 to 48 hours, and also look for an email from the team reaching out to you if you have any more questions, concerns, and their contact information is in the chat. So with that, I'd like to thank you all again for today, and we look forward to reaching out and speaking with you soon. Thank you very much.